We are a pioneer in T-cell therapy, and we invented CARVYKTI, which is now #1 selling CAR T in the world. But our researchers recently had some breakthrough called in vivo CAR T, where we're really moving the factory to inside of the body. So we're injecting the viral vector directly into the vein, and then in the vein, the T cells will be transduced. And then, of course, over time, it will be expanding into a therapeutic dose. So at this EHA, we have some exciting data to share with all the scientists and physicians here. We had actually dosed 12 patients as of clinical cutoff date of April 1st. The first 6 patients were dosed in dose level 1, and then we moved to higher dose. Another 6 patients were dosed in the dose level 2, where we saw all 6 patients responded. So we have 100% overall response rate. And what's more remarkable is that among those 6 patients, actually we had seen already 5 complete responses, where by the means of PET CT scan, you don't see any cancer activities left. So that is 5 out of 6, or 83% of complete response rate. So among the 6 patients in dose level 2, the median follow-up is about 2.2 months. This is very exciting data, but also very early. We realize that. We continue to follow up. Among the 6 patients tested, and that will be reported here, basically you see that the response is up to day 120, so roughly 4 months. We'll continue to follow up, and then we'll answer the question about durability.
Okay. One question I have is the traditional CAR Ts that are on the market today for this particular type of blood cancer target something called CD19. Mm-hmm. In this case, you designed it to target 2 different things, CD19 and CD20. I'm wondering, why did you choose to do that? And does it add an extra, like for in vivo work, did it add an extra technical hurdle to make that possible?
So you're absolutely right. We are targeting both CD19 and also CD20, the 2 types of cancer antigen on the surface of lymphoma cells. The reason being that you already have 3 commercial CAR Ts in the market targeting CD19. However, after a few months, many patients relapse. And based on clinical data, we know that roughly 2/3 of those relapse was driven by what we call downregulation or escape of CD19 antigen. So in order to prevent this kind of relapse and also hopefully improve durability, our scientists designed the CD19 and CD20 dual targeting in vivo CAR T. In this case, it's quite unlikely that the cancer cells will easily develop the mutations against both antigens. That is why we designed this in vivo CAR T targeting both CD19 and CD20. Now, with 12 patient data, it's hard to say whether definitely this is better, but given the early data we're seeing, we're very encouraged by the 100% response rate and also 83% CR rate.